Hi, my name is Morgan Robinson and this is my video on Exam 3. Today I will be explaining the synapsis of chromosomes and meiosis. Synapsis occurs during prophase 1 of meiosis where homologous chromosomes pair and cross over, as you can see in this image. To illustrate the process of synapsis, we will start with individual 1, who will have the chromosomes MLI centromere JKHG and GHI centromere JKLM. Flipping one of the chromosomes to match the ends, we can easily see that the paracentric inversion has occurred. This means that there is an inversion with the segment containing the centromere. In order for this crossover between the J and K regions to occur, an inversion loop is needed. For this specific crossover, the inversion loop would look like so, with each color representing the different paths that can be taken, and an X to represent the crossover between the J and K regions. If you follow all of the different lines, you can see that the 4 chromatids are formed. Out of these, only GHI centromere JKLM and GHJK centromere ILM are viable because there is no missing genetic material. Now we will do another example that we will call individual 2. Individual 2 will have the chromosomes MLI centromere JKHG and GHKJ centromere ILM. Again, if we match up the ends, we can easily see that the one chromosome is basically just the reverse of the other. The crossover between the J and K regions for these chromosomes would look like so, again with the different colors representing the different paths taken, and an X to represent the crossover between the J and K regions. The 4 chromatids formed would look like so, and all are viable as there is no missing genetic material. With this, there is an expected difference in fitness between individual 1 and individual 2. Because all of the gametes produced are viable, individual 2 has a greater expected fitness.